All right. So let's talk again about central dogma. What was central dogma? What did it say? I didn't hear you. What did central dogma say? DNA to RNA and RNA to? Very good. So DNA to RNA is transcription, which we did last time. We also did formation of DNA from DNA, which is replication. And today we will be doing translation from DNA to proteins. Very important part. Everything is important to understand, but this is a little bit complicated. I will try to make it as easy as possible. All right. First, let's understand some vocab words, the main words that we will be using in this whole process of translation. First is the codon. Now, what is a codon? Now, there are 20 different amino acids. We have 20 different amino acids. We all know proteins are made up of amino acids. Now, these amino acids, they combine together to form the proteins. We know that also. So, there are different combination of proteins or amino acids that will join together and they will form the protein molecules, right? Now, these amino acids are recognized by a code language. It is called a codon. So, three bases together form one amino acid. For example, one amino acid is methionine. This is the name of the amino acid. That's called methionine. It is represented by AUG. So this is a codon. It is made up of three nucleotide bases, three bases. We all know that DNA, mRNA, all are made up of four bases. We did those four bases, A, T, G, C for DNA and A, U, G, C for RNA, right? So three bases together will combine to form a codon. Codon means it codes for one amino acid. It's basically, you can say it's the name, there is a name for the amino acid, but the amino acid says, I don't recognize the name. I need you to convert the name into a code which I can understand. So this methionine amino acid says, give me a code. If you give me that code, then I can understand and I can come to you. So the code given to this is AUG. All right, so that's why it is called a codon. It's a code. Everything has their own code language. Sometimes parents want to talk to each other so that the kids don't understand what they're talking. So they use a code language. So this is a code which only the amino acid can understand. All right. So it is basically three nucleotide bases that code for one amino acid. All right. Good. All right. Now, another thing is the transcriptional unit. Now, what is this transcriptional unit? So this is the mRNA. This is the mRNA or RNA on which the protein synthesis will take place. Protein synthesis is from RNA to proteins, right? RNA to protein. So RNA has to take part in protein synthesis. Now this transcriptional unit has all these things in it. What is it? It has a start codon. Start codon is always AUG, which is methionine, as I told you, example. So AUG is the start codon. What does that mean? Like there is an entire mRNA. So where should the protein synthesis start? There has to be some initiation point. There are so many codons, but nobody knows where to start. Like we did in DNA replication, that a primer has to be there, which helps the DNA polymerase. It gives signal. Now I am here. You can start the synthesis of DNA, right? Same way here, there has to be something which says, okay, you can start the protein synthesis from here. So AUG is recognized as a start codon from where the protein synthesis will start to take place. All right. Then we have the stop codon. As the word indicates, when the protein synthesis is taking place, after particular number of amino acids join together to form a protein, the protein synthesis has to stop, right? Then the signal has to be given from somewhere that, okay, we are done. Now we have to stop the protein synthesis. Otherwise, it will keep on synthesizing and anything in excess can lead to cancer or any other happenings in the body, right? So the stop codon, there are 3 stop codons, UAA, UAG, UGA. You don't have to remember them, but just to know, there are 3 stop codons that are fixed. It's also, I just made it like this. A means come, AG, and it's the opposite, UAG and then UGA. They both are opposite to each other. All right, but you don't have to remember this, that's fine. So there are 3 stop codons. Now this is the stop codon here in the end. So when the protein synthesis is taking place, once this part is reached, this codon is reached, the protein synthesis stops. Okay, so there is a stop codon. Then there is UTR. What is UTR? Means untranslated region. So in this whole mRNA, this is the part in between the start codon and the stop codon where the actual mRNA is helping in translation of proteins, or where actually the protein synthesis is taking place. The codes are being read. But here there are some regions in the beginning and in the end which does not take part in protein formation. It is untranslated. Formation of proteins from RNA is called translation. So if we use the vocab terms, it is basically called as this part is being translated into proteins. This part is not being translated into proteins. Or UTR is a part that is not read and it does not form proteins, in simple words. All right. And then we have the coding region, which is this mRNA coding region, which codes for the amino acids. All right. So the transcriptional unit consists of these 4 things. This is the mRNA. In mRNA, we have what do we have here? What do I have here? Yes, start codon, from where the actual synthesis of protein is taking place. Starts, is basically starts here. Stop codon, here the protein synthesis ends, stops. Then UTR, the untranslated region, the region in the front and region in the back. These regions do not take part in the protein formation. They are not translated. They are not coded. And then we have the coding region, the region in the center of the start and the stop codon, where actual coding or the actual synthesis of amino acids or the protein is taking place. All right. So this is all what is present in transcriptional unit. Now, next thing that is important in the formation of proteins is ribosomes. mRNA and ribosomes, they both help together in the formation of proteins. Now, these ribosomes have 2 subunits. You can see this is the larger subunit, and this is the smaller subunit. One big subunit and one smaller subunit. And these ribosomes, they are made up of proteins. You can see these are made up of proteins here and also rRNA. Small r, rRNA. This is also called ribosomal rRNA. All right. So these are made up of 2 subunits. One is the larger subunit, one is the smaller subunit, and these subunits are made up of proteins and rRNA. To make it further clear, there are 3 types of RNA. 3 types of RNA. One is mRNA, which we have been talking about. This is mRNA. It is also called messenger RNA because it carries the messages for the formation of proteins. Okay, it carries the messages for the formation of amino acid, for the formation of proteins. So this is mRNA that is actually used in formation of proteins. Then there is tRNA. tRNA is also called transfer RNA. T is transfer RNA. What does this do? It also helps in protein formation. It basically transfers the amino acid from the cell, the cytoplasm of the cell, in the cytoplasm. All this is happening in the cytoplasm. All this is happening in the cytoplasm. All right. So amino acids that are actually required for the formation of proteins, those are all present in the cytoplasm. So in the cell, let me make it further clear. In the cell, if you remember, this is nucleus and this is all cytoplasm, right? Now this synthesis is taking place in the cytoplasm. This is the mRNA, these are the ribosomes. Now this tRNA is bringing the amino acids from the cytoplasm, all over from the cytoplasm. It transfers the amino acid from the cytoplasm and brings them here to the ribosomes and mRNA. All right. So it is transferring, this tRNA is transferring the amino acid from the cytoplasm and transferring to where? To the mRNA and the ribosome. All right, so that's why it is called transfer RNA. You will understand it better when we'll do the actual whole process. Then rRNA, this is also called ribosomal rRNA, and we just did it. It is a part of large and small subunit of the ribosomes. All right, so these are the three types of RNA. Now, this is the structure of tRNA. tRNA is the transfer RNA. It is also called cloverleaf structure, or it's kind of inverted L-shaped structure. So this cloverleaf structure of tRNA is basically, the important part in this is the anticodon site. Now, what is this anticodon site? So if this is the mRNA, this is this mRNA, and it has this codon AUG. It has this codon AUG. Now this is the codon. Now the amino acid has to be brought here. Now this codon is the code for methionine amino acid. So it is giving a call to methionine amino acid. But this amino acid are all the way in the whole cytoplasm. Who is going to get these amino acid from the cytoplasm? To this mRNA. This is the mRNA. This tRNA says, okay, I'll help you. I'll go and find out the amino acid which is opposite or the methionine amino acid, which codes for AUG. Now, how it is going to search? So it goes and searches in the whole cytoplasm, brings the methionine amino acid on its arm like this. It carries the methionine amino acid in its arm and it brings to the mRNA. And it says, see, I got your amino acid. But the mRNA says, how do I believe you? I need to check it. Now, how does it check? So on the opposite side, it should match with the mRNA. It should be the base pairing rule. It should be the anticodon. So if this is the codon AUG, what should be on the other side? If this is A, it codes for U, because we are talking about RNA. We are not talking about DNA. So it will be U, not T. Remember, A codes with U and G with C for RNA. In DNA, this is T. but we are talking about RNA. So this will be U, right? So if this is A, this should be U. If this is U, it should be A. And if it is G, then this should be C. All right. So this is the anticodon for this codon. Got it? So now when this tRNA comes, it says that I have your methionine amino acid with me. Then the mRNA says, come here, I have to first check it. And it checks with this, its anticodon. This anticodon is fit in with this codon. And now it says, okay, we are fine. And then it accepts this methionine amino acid. All right. So this tRNA plays a very important role in bringing the amino acids, finding the amino acid in the cytoplasm of the cell, and it brings it to the mRNA. This is the mRNA, and these are the ribosomes on top of it. We'll do this structure in detail. All right. So this tRNA has this anticodon arm, you can say, or anticodon loop, you can say, which is very important for the recognition of the amino acid. And this is the amino acid binding site where actually the amino acid attaches itself. All right. Now, one more thing in the ribosomes. We did the ribosome has a big subunit and a smaller subunit, right? Now, this larger subunit has three parts, EPA. It has three doors, you can say. EPA, you can learn it as EPA. All right. Now, this is the A site. For the entry of charged tRNA, I'll tell you this in detail. Just to know for now, there are three doors, A, P, and E. This is the A site. This is the P site, where actually the tRNA containing the growing peptide comes and launches itself here. I'll just tell you. And this is the exit site from where the tRNA exits out. So there are the three sites or the three doors on the larger subunit of the ribosome. All right. Now, let me just talk about analogy of this whole process so that it becomes a little bit easy to comprehend what's going on. So now, for example, we start with the DNA. Okay, so we are considering the whole process of this as making up of cookies. You are making the cookies in the kitchen. How do we correlate that to the process of transcription and translation? Our cookies are the final product, which are the proteins. All right. So if we start with the recipe book, now we have a recipe book. From that recipe book, the whole book, we just want one recipe of making the cookies. All right. So we have to copy down that recipe and then we have to take it to the kitchen. Okay. So that recipe book, the whole book is the DNA. We have to copy it down with the help of the pencil. We have to copy the recipe down with the help of the pencil, right? So we are copying the DNA to make mRNA. All right. And with the pencil, you remember we did RNA polymerase, the enzyme which was responsible for the formation of mRNA from DNA. All right. So this is basically the whole recipe book. The whole recipe is our DNA. From that, we are copying just one recipe. So this is the process from making DNA to RNA with the help of RNA polymerase. And what is this called? Transcription. Okay. Now what happens? Once we have the recipe, we take the recipe in the kitchen because we have to make the cookies. So when we take the recipe in the kitchen is the cytoplasm. If you remember, DNA to RNA was taking place in the nucleus of the cell. Once the mRNA is formed, it comes into the cytoplasm of the cell. All right. So once this is done, it comes to the cytoplasm in the actual kitchen where actually the formation of cookies will take place. Now in the kitchen, we have the stove. Now what is the stove? There. Stove is very important because actually we have to mix certain ingredients in that. We have to warm certain things in that. So those are basically the ribosomes. We consider those to be our ribosomes. Then when we are making the cookies, what are the cookies? Cookies are actually the proteins, the final product. And we need certain ingredients for making the cookies, right? What are the ingredients? So when you go in the kitchen, you need the ingredients. You need to collect the ingredients. What are the ingredients for making the cookies or for making the proteins? What is it made up of? Amino acids, right? So amino acids becomes the ingredients. And finally, who is doing all the work? Moving around, bringing the ingredients to one place, working on the oven, working on the stove. Who is doing all that work? You. You are the chef. So who is moving around? It's the tRNA. The transfer RNA is the chef, that is you. So if we imprint the whole thing one more time, we want to make cookies in the kitchen. So we have the whole recipe book with us, but we need just one recipe. So from the whole recipe book, we copy down just one recipe for making the cookies. So the recipe book is the DNA. Copying down with the pencil is the RNA polymerase. And the single recipe that we have now is the mRNA. Now we take that recipe to the kitchen. The kitchen becomes the cytoplasm. Now in the kitchen, we need the ingredients to start working with. What are the ingredients for making the cookies? Cookies are the actual proteins. What are the ingredients? Amino acids. And who is working here and there to gather all the products? You are the chef. So you are the tRNA who is moving around to get the stuff. And where actually all this happening is in the through the ribosomes, which you can say the oven or you can say the stove, whatever you want to say. All right. So this is the simple analogy which will help you to understand things better. All right. You can relate it more. Okay, now the actual process of translation. How does that take place? So we know the basics, all the small basics that are involved in it. So it has 3 steps: initiation, elongation, and then finally termination. So let's take this as the mRNA transcript or the translation transcript that we talk about this, their transcription unit that we have here. Now here, first of all, the smaller subunit of ribosomes will come and bind to this transcription unit. And then you have the start codon over here. You see this is UTR, some untranslated region. Then we have AUG, which is the start codon. So the tRNA with methionine amino acid, which is the initiator tRNA. tRNA will bring the methionine, which is the starting point or the starter amino acid that we talked about there, the start codon. It will come and stick to this AUG. Okay, the tRNA that carries methionine, which is the starting point or the start codon. The tRNA comes, searches everywhere in the cytoplasm and brings methionine and comes and attaches over here, which is AUG. This is the untranslated region. All right. So this is here, the initiator tRNA will come and bind to it. This is the initiation. Then what will happen is, after this is started, then another tRNA will come. Now this is the next codon, GUC. Now tRNA will go and search for another amino acid in the cytoplasm pool, which will have the anticodon for GUC. For GUC, what will be the anticodon? For G, it is C. For U, it is A. For C, it is G. So it is going to search for the amino acid that is going to code for this, and it is going to bring here. So these are the 3 sites, and then the large subunit will come and join here. So this is the smaller subunit of RNA. Initially, or in the initiation part, the starting part, the smaller subunit of RNA will come and join. Then the initiated tRNA, or the tRNA that starts the process of translation, which is methionine, will come and attach to the site or the start codon. Then the larger subunit of this ribosome will come and join. Okay? Now the larger subunit comes and joins. So if you remember, we did 3, there are 3 doors in the larger subunits. This is E, this is P, and this is A. Okay? Now this methionine is here. These 2 sites are empty, E and A. So on A site, another tRNA will come, which has the anticodon for GUC. It will go and search for the amino acid, and it will come and bind here. So A site is the site where the new tRNA will come. Okay, this is the new tRNA which has the amino acid, which has the anticodon opposite to this. Right? Now the this tRNA which has the amino acid is called the charged tRNA. Charged. The tRNA with amino acid is called charged tRNA. Now this is the charged tRNA. This is also charged tRNA. Now what will happen? This one tRNA at the P site will say, see, now I have grown old. I don't want to do my work anymore. You take the burden of this one, of this amino acid which I am carrying. I cannot carry it anymore. I want to exit the game. So it says that you take my amino acid from me, and it forms a bond with this. So what happens? They both form a bond. This comes over here. This is left alone. It does not have any amino acid on it now. Now this is called uncharged tRNA. It is called uncharged tRNA now. Okay, so what will happen? Now this tRNA over here will have 2 amino acids, and this will be uncharged. Once this becomes uncharged, this whole mRNA will move further. You can see here. The whole unit, it moves further. So mRNA moves further. So this whole ribosomes shift. You can see here. From UTR, the whole ribosomes have made a shift. So now what will happen? Till now it was EPA here. It was at UTR, AUG, and GUC. Right? Now what will happen? The ribosome moves. The whole ribosome moves. Now, because it says that I am old, I don't want this anymore. I want to exit the game. So ribosome also has to make some changes then. It says, okay, hold on, let me move. It moves 1 step or 1 codon. Now you see, when it moves 1 codon, everything moves. When the EPA site moves. So what happens now? This uncharged tRNA at AUG, it comes, till now it was at P site. It was at P site. Because the ribosome moved, now it will come at the E site. Now it will come at the E site. You see AUG? AUG is still here because the ribosome has moved. From the P site, it comes to the E site now. This is EPA. This tRNA or this tRNA now has 2 amino acids because it has transferred its amino acid till here. It has 2 amino acids, right? Now it has 2 amino acids over here. Since ribosome has moved, so now this A will come at P. This A position will come at P position. So GUC, GUC. But now this position from A has moved to P. And this position becomes empty. This A becomes empty. This becomes empty. So now what will happen on this empty side? New tRNA will come. A new tRNA which has the anticodon for UGC, they will come and attach to here with the new amino acid. Okay, so this is A site, P site, and E site. Once again, so what is happening in the whole thing is at first the smaller subunit of ribosomes attach. The first tRNA comes, which is the carrying the methionine, which is the initiator or starter of the whole process. So this tRNA comes with methionine and the process now starts. This is the initiation. Then the larger subunit of ribosomes comes and bind over here. When the larger subunit bind over here, there are three sites, E, P, and A. Now on this side, the P side, we already had this methionine over here, right? Now this one, at this site, another tRNA comes, which has an anticodon for GUC. It brings another amino acid. All right. Now what happens is this tRNA says, I want to exit the game, I'm too old, and you are new, you carry my burden. The ribosome says, okay, hold on, I have to move too. So now this ribosome makes one shift. When this ribosome makes one shift, the three positions also shift automatically. All the three doors are going to make a shift. So this EPA was here from UTR, but now it has made from a shift from UTR. So now this side becomes EPA. Now what happens? This tRNA, which had the methionine, it had transferred this methionine to this tRNA, right? So now it does not have any amino acid on top of it. So this is called uncharged tRNA. This one has two amino acid. One is methionine and one is its own amino acid. So now when the ribosome has moved, this uncharged tRNA will move to the E site, one side shift. This tRNA will have two amino acid, will move to the P site, and this site will be empty, A site, where a new tRNA or the new charged tRNA will come into place. Same thing will happen. So now this is uncharged tRNA, which has given its amino acid to this. This is going to exit the game. This is going to leave this. All right. Then again, what will happen? It has two amino acids. This is going to say, see, I have become old. I don't want to take part in the game. You take my burden. The new one, the new amino acid. So this is going to come to here again. Again, the ribosome will move. When the ribosome will move, this will become empty because it has transferred both the amino acids to this one. This will get two more amino acid. This will become empty again. This has already exited the game. So now again, it moves like this. So what will happen? This will be, the uncharged tRNA will be on the E position. This tRNA with the growing polypeptide chain will be on P position, and this will be empty. So here again, the new tRNA will come, which has the anticodon for GCC. Now this new tRNA will come. Again, the same process will repeat. This is the A site. So now you see the A site was for the entry of charged tRNA. This is the point of entry for the new tRNA. This is the point where the polypeptide is growing, P. E is the site where the exit of the uncharged tRNA is taking place, or the tRNA which has no amino acid on top of it is taking place. All right. So this what keeps on happening. The ribosomes keeps on moving. This new tRNA comes at A position, EPA. The new tRNA comes at the A position. Then the one at the P position says, I am old. I don't want to take part in the game. You take all my amino acids. So all the amino acids are transferred here. This becomes empty, or you can say uncharged. Then the ribosome says, wait, I will give you a place to exit the game. Otherwise, where you will exit the game from. So the whole thing shifts. Ribosomes make one shift. This uncharged tRNA comes on E position, exit position, and it exit the game. Now this tRNA at the P position has all the amino acid, and again A site is empty where the new tRNA comes into place, the charged tRNA. All right, and this continues till it comes to the finally stop codon. Now here at the stop codon, there is a long polypeptide chain at the EPA site. So here at the A site, no new tRNA will come. There is no new tRNA comes and binds to this site because it's in the untranslated region. Okay, no new tRNA will come and bind here. There is a releasing factor that comes and attaches here. When the releasing factor comes and attaches, this polypeptide is released and the two subunits are released as well. And this is the end of translation. All right, towards the end, this is called termination. In the termination process, only the releasing factor comes at the A site. Once the releasing factor comes, the polypeptide is released. You can see this polypeptide is released and both the subunits of RNA is released. All right, so this is the whole process of translation. Do you want me to go through it one more time? Or you're good? All right. So how do we start this process? This is the mRNA. mRNA has untranslated region on the either side. This is the AUG, which is the start codon. First of all, the smaller subunit of ribosomes will come and attach. Once it attaches itself, the initiator tRNA means that initiates the process, will go into the cytoplasm. It's going to find methionine. Methionine is the start codon. Methionine is the amino acid that starts the whole process. So it's going to search for methionine amino acid. It's going to bring it from the cytoplasm. And from the cytoplasm, it's going to come here on the mRNA. That this is the tRNA with the methionine. You see, this is the tRNA with the methionine and the anticodon. So it comes here and it attaches itself here. Now, what will happen? The larger subunit of the ribosomes come and attach over here. Now it has three sites or three doors, EPA. Okay. Now here the methionine is attached. Here the methionine is attached. Okay. This is empty right now. So what is going to happen now? So this is empty. Only methionine is attached here. So this is the A site which accepts the new tRNA. New tRNA, which are the charged tRNA. New charged tRNA. Charged means they have amino acid attached to them. All right, they have amino acid attached to them. So now here, opposite to this, GUC, another tRNA will come, which has the anticodon for GUC, and it will have its another amino acid. It will come and attach here. Okay, this is the A site now. Now, this one will say that at the one at the P site, I am done with the game. I am old. I want to exit the game. Can you please accept my amino acid? So the one at the A site will say, okay, I will accept it and you can exit the game. But ribosome say, hold on, I need to provide you a place from where you can exit the game. So this ribosome, it is going to move. It is going to shift from one place or one codon. So it moves from UTR to AUG. AUG, GUC, and UGC, this whole place. Now, when it moves from here to here, then what happens? Now, what will be the position of each of them? Again, E, P, A. Now, what will happen? This will come at the E position when it moves. And now this is uncharged. This is called uncharged tRNA because it does not have methionine on top of it. It has transferred to this one. This one, now this one at A position will come at P position. Now this one carries two amino acids on top of it. Right? This is P site, which has the growing polypeptide chain. that we did over there. This is the growing polypeptide chain. And now A site becomes empty again. So now this A site will accept another newly charged tRNA. So it is going, tRNA is going to go and search in the cytoplasm. It is going to bring another tRNA, which is opposite to GCC. All right, another tRNA will come to this position. Same thing will repeat. The tRNA over here in the middle at the P point will say, I am done with the game. I want to exit the game. Can you accept my these? What do you call these amino acid? It is going to say yes. So these are going to transfer here. It is going to come to this position at the E position, and then it is going to exit. This growing polypeptide chain will come here, and this position will become empty again to accept new tRNA. So this process is going to continue till they reach the stop codon. Once the stop codon is reached, which are 3 stop codons, once the stop codon is reached, this A site will not accept any new charged tRNA. It is going to say, I'm done with it. Now the releasing factor will come. And once the releasing factor comes here, this polypeptide chain is released and both the subunits are released and this amino acid or the protein is formed. All right. Lot many things going on, but if you understand it, you will be able to go through it very easily. All right. Any problem, anybody, any questions?